Hello everyone. Today we'll be talking about apoptosis. Apoptosis is basically a type of cell death. To be more specific, it is a tightly regulated suicide program, or also known as programmed cell death. Now, when I say that this is a tightly regulated suicide program, it is important to note that this is regulated by a small number of genes. And also note that this apoptosis, this type of cell death, can be both physiological as well as zoological. Now, if I have to give you examples, I'll split them into two groups, that is physiological and pathological. Physiological, you have all your embryological examples such as the apoptosis of the notochord or the thyroglossal tract or the thymus or whatnot. Tract, the thymus, etc. Or you can say a removal of self-reacting T cells. There's many, like hormonal dependent involution or removal of excess cells post-inflammation. There's many. Now, when it comes to pathological, you have to note three important things. Is that it is first seen in viral infections in case of DNA damage or intracellular accumulation of misfolded protein. Intracellular accumulation proteins, which is seen in diseases like Alzheimer's. Now, coming to the actual mechanism of apoptosis, apoptosis has two phases. Number one is the initiation phase. This initiation phase has two pathways, the extrinsic pathway, which is seen in case of viral infections, and it is mediated by caspase 8 and 10. We'll discuss about caspase soon. And there is intrinsic, which is seen in case of DNA damage or protein accumulation. And this is mediated by caspase 9. And both pathways, whether it be extrinsic or intrinsic, will end in a common execution pathway. Execution pathway or phase. Both will end in a common execution phase. Now, apoptosis is basically mediated by caspases. Now, I'll discuss about caspase here itself. As the name suggests, caspases here, C stands for cysteine, which is present at the active site. And ASP stands for aspartic acid. So basically, it is a protease with cysteine at the active site, which cleaves after aspartic acid. That is all. Now let's get into the initiation phase proper. In that, first we'll be discussing about extrinsic pathway, which I said happens in the case of viral infections, and it is mediated by caspase 8 and 10. Now before I start with the extrinsic pathway, I like to take an example of a normal cell. This is a normal cell and this is the nucleus. So in case of a normal cell, there are no death receptors. We'll discuss about death receptors very shortly. So there is no initiation of the entire apoptotic pathway. Now, in case of, let's say there's a viral infection, this is the cell, this is the nucleus, and say there is a viral infection, there'll be increased expression of these things called death receptors. They're called FAS or death receptors in case of a viral infection. Now, let's see how this death receptors act. Okay, so as I said, there'll be something called death receptors or the FAS receptor. This is the FAS receptor. FAS receptor, and every receptor has a ligand. So when FAS receptors are expressed, the FAS ligand will come and bind to the FAS receptor. This is the FAS ligand which is present on this T cell. So this will bind to the FAS receptor, which will activate FAS, which will then activate something called as FAD. FAD stands for FAS associated death domain. This is pretty simple. Now what this FAD will do is it will activate pro-caspase 8 and 10 into caspase 8 and 10. That's all. The extrinsic pathway is comparatively simpler. By simpler, I mean the intrinsic pathway has a lot of proteins to mug up. So now if we have to discuss the intrinsic pathway, coming to intrinsic pathway, which is also known as the mitochondrial pathway. Now this pathway is regulated by some special proteins like the pro-apoptotic proteins, anti-apoptotic proteins, and sensors of cell stress. Now all these proteins are basically ion carriers, ion channels. Now you can guess that normally in a healthy functioning cell, anti-apoptotic proteins are active, as in they prevent apoptosis. Of course, you don't want a healthy cell to undergo apoptosis. So way to remember it is that anti-apoptotic proteins give a long life to the cell. L for long life, and all these anti-apoptotic proteins have L in their name, like PCL2, PCLXL, and MCL1. That's just a tip to remember the names. Now, pro-apoptotic are BAX and BAC. Sensors of cell stress are BIM, BAD, PID, PUMA, NOXA. Now, let's go to the mechanism of the intrinsic pathway. Here again, I'll take an example of a normal functioning cell, mitochondria in fact. Imagine this is the mitochondria. And we know that the function of mitochondria is to form ATP. This is normal mitochondria. And to form this ATP, it needs this special protein called cytochrome C. cytochrome C helps in the production of ATP. So normally you would want the cytochrome C to be in the mitochondria itself. You don't want it to leak out. If it leaks out, the mitochondria can't form ATP and the cell would die. So normally the protein which is active or which is present on the mitochondria is this BCL2. As we discussed previously, it's anti-apoptotic. So if BCL2 is active, which is anti-apoptotic, it prevents exit of cytochrome C. Now, if cytochrome C doesn't exit into the cytoplasm, it will remain in the mitochondria, and mitochondria can normally generate ATP, normal ATP generation. So, if ATP generation is normal, the cell function is normal, there will be no apoptosis. This is in case of a normal mitochondria. Now, let's assume there's some sort of stress. For example, it can be, say, a DNA mutation. This cell stress is sensed by the sensors of cell stress, which were BIM, PAD, PID, PUMA, and NOXA. So this will be sensed by BIM and PID, and this stress increases the expression of these sensors of cell stress, and these will do two things. One, it will destroy the anti-apoptotic proteins like the BCL2, and it will activate pro-apoptotic proteins like PAX and BAC. As I already said before, all these proteins are ion channels. So these pro-apoptotic proteins, which are ion channels, will allow the exit of mitochondrial proteins like cytochrome C. So cytochrome C exits into cytoplasm. There, along with something called as APAF1, which stands for Apoptosis activating factor 1. Apoptosis activating factor 1. So cytochrome C in the cytoplasm along with apoptosis activating factor 1 will form a multimeric protein called the apoptosome. Now this apoptosome, what it does is it will activate procaspase 9 to caspase 9, and along with the exit of cytochrome C, there is also exit of weirdly named proteins called MAC and Diablo. Basically, these act as physiological inhibitors of apoptosis. So that was all about the initiation phase. As I said, both the pathways, whether it be extrinsic or intrinsic, will end in a common execution pathway. So let's talk about the execution phase. So by the end of initiation phase, we have activated caspases 8, 9, and 10. It will do two things. Firstly, it will activate caspase 3 and 6. Yes, caspase 8, 9, and 10 will further activate caspase 3 and 6, which are basically proteases also. Now, this will digest the intracellular protein content. So, say this was the cell before. This is the cell, this is the nucleus. When the protein gets digested, the cell size will decrease, firstly, and after protein digestion, the cell will appear deeply eosinophilic. It will appear dark pink. So the cell would be smaller or shrunken, and it will appear dark pink. And other than that, caspase 8, 9, 10 also activates another enzyme called endonuclease. This endonuclease will, what it does is it will cut the DNA into fragments at specific sites. So there will be chromatin condensations present. And this chromatin condensations is the hallmark of apoptosis. Now, to summarize, by the end of the execution phase, three major things happen. That is, okay, let's assume this was the cell before apoptosis, and this is the nucleus. After execution, the cells would be smaller in size, the nucleus will be condensed, and it will be deeply pink, dark pink, eosinophilic. So when a cell undergoes execution phase, it forms something called apoptotic body, which is smaller in size, which has a dark pink cytoplasm, and a condensed nuclei. Now, this apoptotic body has to be removed from the system also. It is removed by macrophages. The special thing is that this process is a silent eating process. In the sense, there'll be no inflammation, and there'll be no pain. This special form of eating is known as apoptosis. A few words about apoptosis. What happens in apoptosis is that the apoptotic body is broken down into tiny pieces, which is eaten by macrophages. And the opsonins which are involved are, firstly, there is phosphatidylserine, and there is also something called as thrombospondin. Now, Normally, these are present on the inner aspect of the cell. Inner aspect of the cell. In case of apoptosis, what happens is that these are flipped outside. So they are now present on the outer membrane, and these give off something called as eat me signals. And these signals are sensed by macrophages, which will come and clear all these apoptotic bodies. Now that was it with apoptosis. Lastly, how would you detect, how would you say that apoptosis has taken place? The next and the last topic is detection of apoptosis. Can be done by things like, number one, as I said, if the intrinsic pathway takes place, there will be exit of cytochrome C into the cytoplasm. There will be increased cytochrome C levels in cytoplasm. And in case of microscopy under HND, you'll be able to see apoptotic bodies. I'll mention again, the apoptotic bodies are small, dark pink with condensed nuclei. Yes. And other than that, you can check the DNA gel electrophoresis. In DNA gel electrophoresis, you will note that this is the, normally you would only see DNA in one place. In case of apoptosis, the DNA will be fragmented, as I said. There will be a step ladder pattern. In case of necrosis, since the DNA itself gets lysed, there will be a smearing pattern. So this is normal. This is a step ladder pattern in apoptosis. And this is smearing pattern in necrosis. In apoptosis, there will be DNA fragments. And in case of necrosis, there will be lysed DNA. So these are the images. Here you can see this small dark pink with condensed nuclei. This is the apoptotic body. And in DNA gel electrophoresis, this you have A, B, C. A is normal and B is the step ladder pattern. Which is seen in apoptosis. And C is the smearing pattern. Seen in necrosis. So that was all with apoptosis. Thank you for watching.